Hello. So, continuing to study session 4, which I think is titled fatty acid oxidation. Well, from my chemical point of view, I don't suppose I'm going to approach it like this, but of course, this is a special case. So, we've handled glycolysis and the citric acid cycle, or what others called the tricarboxylic acid cycle. Now, how do you hydrolyze fatty acids, right? I mean, you still remember your classes of food. Yeah. Here I think I can see here in Africa, we eat a lot of carbohydrate-based diets, right? But this, for example, burger is an example of a food that is rich in fat, sugar, and salt. Fat spread of food, for example, butter, oil, things like that. So if you look at this pyramid here, it's recommended that you take less of these two foods, right? Fats, food that are very rich in fat. You need them, but not too much. And taking too much, consuming too much of those is what's causing the case of obesity and cardiovascular diseases in the developed world, right? You need proteins. You need more proteins than fat. Of course, your food should be rich in proteins. Should eat chicken every day if possible. Okay. 2 servings a day, according to this pyramid. Do you eat chicken 2 servings a day? Eggs and nuts. Should eat 2 servings a day. Okay. I mean, it's almost a crime if you don't eat chicken 2 times a day. I mean, you should eat chicken, milk, yogurt, and cheese at least 3 times a day. After eating, you take yogurt as dessert, cake, right? Cheese, sorry, not cake, cheese, yogurts, the thick yogurts and the thin one as well. Okay. So your whole meal, cereals, breads, of course, we eat a lot of cereals, rice, potato, bread. Of course, although bread is quite expensive now, not many people eat bread, to be honest, but of course, you know what I mean, right? And then your vegetables. Now let's go back to our lecture, fatty acid oxidation. We've talked about glycolysis, the oxidation of glucose through the glycolytic pathway and the citric acid cycle. So fatty acids serve as a more efficient source of energy than carbohydrates. Why? Because they are reduced and anhydrous. Looking at the structure, you can go and browse the structure of carbohydrate and fatty acid compare. Carbohydrates are more hydrated, while fatty acids are more reduced. They don't have molecules of OH, not much molecules of OH, hydroxyl functional group compared to the carbohydrates. They are anhydrous. So the energy yield from 1 gram of fatty acid, this is the reason now. The energy yield, why is fatty acid a better source of energy? It's because the energy yield from 1 gram of fatty acid is approximately 9 kilocalorie compared to 4 kilocalorie for carbohydrates. CHOS here means carbohydrates. So basically that's the reason. Carried up the rest, but I think basically I've communicated what we needed to communicate here. All right. So utilization of fatty acids for energy production varies significantly from tissue to tissue. Of course, some tissues will prefer, depending really. Let's just give an example. Like they are a major source of energy in cardiac and skeletal muscle. That's fatty acids. While the brain utilizes them poorly, the brain is not a big fan of using fatty acid as a source of energy, right? So they prefer glucose, sugar, glucose, for example. And in the absence of glucose, the brain uses ketone bodies, which we are going to discuss in the next module. I think session 1 of module 2, study session 1 of module 2. In addition, the red blood cells cannot oxidize fat. because they lack mitochondria. The oxidation of fat goes through a pathway called beta-oxidation. So they lack mitochondria. So during prolonged fasting, the liver converts acetyl-CoA generated by fatty acid oxidation and amino acid breakdown to ketone bodies, which becomes a major fuel for the brain and the RBC. Okay. Yeah, there's a talk about the activation of fatty acid, which you should read. I'm going to explain using a slide to save time. And the enzymes involved in this catalysis are also mentioned here, which I'm also going to explain. Carnitine acyl transferase 1, carnitine acyl translocase, okay, and what have you. All right, there's an MTQ here. Mention a reason why fatty acids have more efficient energy than carbohydrate. Of course, the energy yield from 1 gram of fatty acid is approximately 9 calories. That's more than twice the energy you get from 1 gram of carbohydrate. 9 divided by 4 is 2 point something, right? So it makes sense, doesn't it? All right. Let's come here. So this was the same question that was posed. Why do fatty acids have more energy than carbohydrates? Because lipids include a high percentage of CH bonds, which store chemical potential energy more effectively than other molecules. So 1 gram of fatty acid gives you 9 kilocalorie of energy, while 1 gram of carbohydrates gives you 4 kilocalorie. Basically, that's the answer from your lecture note. Now, the mobilization of fats. For the fatty acid to be oxidized, they have to be mobilized in the first place, right? Now, usually, when you ingest fats into your system, it goes into the body, your bile salts from the liver, right? Emulsify dietary fats in the small intestine, forming mixed micelles. Bile salts usually secreted, I think, from the gallbladder, right? Okay, and then the intestinal lipases. Lipases are enzymes that act on fats, degrade triacylglycerols. Fatty acids and other breakdown products are taken up by the intestinal mucosa and converted into triacylglycerols. Your triacylglycerols now are incorporate with cholesterol and apolipoproteins. These are proteins. And now this incorporation forms what we call chylomicrons. Chylomicrons move through the lymphatic system and bloodstream to tissues. Lipoprotein lipase activated by ApoC2 in the capillary converts triacylglycerols to fatty acids and glycerols. And this is how you get your fatty acid. You eat fats, and this process results in the generation of fatty acid through the action of lipases, hydrolytic enzymes that break down fats into fatty acids. Now, this fatty acid will go into the cell and through a pathway called beta-oxidation to yield an ATP. It's the energy currency of the cell. Now, this is present in your lecture notes. So this slide is very, very important, okay? The mobilization of triacylglycerols. So the hormones that are responsible for this mobilization, glucagon, right, is a hormone in the blood. Glucagon is released when there's a need for the body, right, to produce glucose. I can be wrong. Please try to verify that. Okay, glucagon is a hormone. It binds to this receptor, right? The receptor acts through a second messenger, the coupled G protein. This is a protein, G protein, coupled to adenylyl cyclase. Now, this enzyme plays a role in the production of this C cyclic AMP. This is a second messenger. This second messenger is doing the work of glucagon. Glucagon cannot go into the cell itself and do the job all it does is bind to a receptor on the cell, and this receptor will send a message that we need a second messenger to do this job. As the glucose level is low in the body, the activity of glucagon will make sure as glucose is being produced by cells, maybe through gluconeogenesis, it will hint to the cell that we need energy, and the energy will try to produce ATP through oxidation of fats repository in your body, fat deposit in your body, right? Okay, so this cyclic AMP activates a protein called protein kinase A. This protein kinase A is a phosphorylating protein. It will phosphorylate your hormone-sensitive lipase. Phosphorylation of hormone-sensitive lipase here activates it. And activation of hormone-sensitive lipase, in addition to other events that will happen here, will lead to the mobilization of your triacyl glycerol, which is your fat basically, right? Fat A triacyl glycerol is now broken down to diacyl glycerol, which is broken down to monoacyl glycerol, and then which is converted to fatty acids, right? And these fatty acids are what you need for the oxidation, beta oxidation here to produce ATP. Does it make sense? I think it does, right? This is very beautiful. It makes sense, and it makes it easier to understand. As you are reading your lecture note now and listening to this, I think it makes sense, right? Now, the issue is the fatty acids will probably have variable lengths, right? Some might be short and some might be long. Short chain fatty acids and long chain fatty acids. Now, how do we reconcile this? The fatty acid with chain length of 12 or fewer carbons enter the mitochondria because beta oxidation of fatty acids occur in the mitochondria, right? So the oxidation, the fatty acids have to be able to reach the mitochondria before beta oxidation occur. Now, if the fatty acid have a length, a carbon length of 12 or fewer or lower than that, they will enter the mitochondria without any help, okay? They can easily go through the membranes of the mitochondria, pass through the membrane transporters and just go. But if pass through the membranes, sorry, without the help of any membrane transporter. But if the chain length, those with 14 or more carbons, if the chain length is more than 12, we are talking about 14 or more, right? Which constitute the majority of free fatty acids obtained in the diet, then there's a need for a membrane transporter, something that will help the fatty acid to cross the mitochondrial membrane and reach the mitochondria. And this is where the concept of the carnitine shuttle comes in, okay? Through the mitochondrial membrane, they must first undergo the three enzymatic reactions of the carnitine shuttle. Now, this shuttle, just like your shuttle, a car that shuttles you from Zaria to Kaduna. Now, this shuttle is going to shuttle fatty acid into the mitochondria because it's too big. You understand? So the first reaction is catalyzed by a family of isozymes. The different isozymes specific for fatty acids having short intermediates or long chain or long carbon chains, for example. Basically, this is your carnitine shuttle here. And the enzymes that are involved in the carnitine shuttle are also shown here, of course. This is your fatty acid. You can see it, right? This is your mitochondria. This is your fatty acid, right? So carnitine acyl transferase 1, carnitine here is going to bind to your fatty acid and then pass through the intermembrane space. This is your outer mitochondrial membrane. You need your fatty acid to reach the inner mitochondrial membrane before beta oxidation will occur, right? So your fatty acid is already in the outer mitochondrial membrane. The carnitine will carry it through the intermembrane space, and it will be in. Okay, sir. Okay. So it will it will pass through the inner intermembrane space and then into the inner mitochondrial membrane through this membrane transporter, membrane, sorry. The carnitine is the membrane transporter, basically, at this stage. Now, this is the membrane. The carnitine, sorry, is the vehicle, and this is the membrane transporter where the carnitine will have to go through. So uh, this port here recognizes carnitine. And without the help of carnitine, this fatty acid, because of its size, cannot pass through on its own. So the moment the fatty acid enters the inner membrane of the mitochondria, here, this is your mitochondrial matrix, right? Carnitine acyltransferase 2, and the carnitine is released from the fatty acid, and your fatty acid now is free inside for the, what do you call it, for the beta oxidation to occur. So carnitine acyltransferase 1 transfers your fatty acid from the outer mitochondrial membrane into the inner mitochondrial membrane, while carnitine acyltransferase 2 now transports the carnitine back. You see this arrow, right? To transport it back outside now because it's free. It has done its job, release the fatty acid. Now this enzyme, carnitine acyltransferase 2, will now transport it back out from the inner mitochondrial membrane into the outer mitochondrial membrane, where it will carry out the action of transporting another heavy fatty acid again into, you understand? So it's a cyclic process. Okay, so now we'll have to talk about the beta oxidation in itself. We've talked about how the fatty acids, the fats are mobilized, broken down into fatty acids, transported from the outer mitochondrial membrane into the inner mitochondrial membrane, where the beta oxidation will occur, right? Now, the beta oxidation is what actually produces energy from fatty acids, the series of reactions that occur, which I believe are 4 steps, really. Okay, these are the steps for the beta oxidation, which of course I encourage you to read. I'll use my slide to just explain. And it's not going to be too different from what you have here. Everything is going to make sense by the end of it. Okay? Okay, so let's go back to my slide. Now, basically, this is your beta oxidation. Beta oxidation leads to the production of acetyl-CoA. Acetyl-CoA now goes through the TCA cycle, which you've done already, citric acid cycle, to produce ATP, right? Respiratory electron transport chain, your coenzymes NADH and FADH2 go through the. So let's count 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. This is a 16 carbon fatty acid. It's called, and it's saturated. So this is palmitate, called palmitate. It's a fatty acid, 16 carbon palmitic acid. Now, the cycle of beta oxidation, 16 carbons will give you 8 molecules of acetyl-CoA. Acetyl-CoA, one acetyl-CoA has 2 carbon atoms, right? So it makes sense. 8 times 2 is 16. So cycles of beta oxidation will give you this, and this 8 acetyl-CoA now will go into your TCA cycle to produce energy. Now, when you have low glucose in your system, this is what happens. The body will convert fatty acid into energy currency for survival. The world operates on the principle of maximum economy, right? This is the beta oxidation of palmitoyl-CoA. Palmitic acid is converted to palmitoyl-CoA, okay? Sorry, there's some comment. So before I take you through this, I think I should mention the steps. You have 4 steps in your beta oxidation. The first step is an oxidation step, right? The oxidation step, which I'm going to explain. The second step is a hydration step. The third step is another oxidation step. And the fourth step is a dialysis step, which leads to the production of acetyl-CoA. Okay. All right. Now, okay. Now, the first step is oxidation, right? Second step, hydration. Third step, oxidation. Fourth step, thiolysis, if you remember. Okay, this is your first step. This is a 16 carbon, uh, 16 carbon fatty acid. Beta oxidation occurs at the alpha carbon. This is your functional group. Alpha carbon, beta carbon, gamma carbon. The last carbon on the left-hand side is called the omega carbon. Now, the oxidation step here is the oxidation of this palmitoyl-CoA to trans-enoyl-CoA, trans-delta-2 enoyl-CoA. Now, this is oxidized to this, while this coenzyme here, FAD, is reduced to FADH2. This is also an energy currency because it will go into the electron transport chain. If you remember very well from the citric acid cycle, the enzyme that does this is acyl-CoA dehydrogenase. Removal of hydrogen is dehydrogenase, and that's an oxidation reaction. Trans delta 2 enoyl CoA is hydrated now. Dehydration, the second stage, right? Hydratase, enoyl CoA hydratase will hydrate this substrate and produce L-beta hydroxyacyl CoA. Now we've had the first oxidation and the second hydration. Now the third oxidation will be now the conversion of L-beta hydroxyacyl CoA, the oxidation of this to beta ketoacyl CoA. If you notice, this is only occurring with the two carbons here, right? Two carbons here close to the functional group. That's what beta oxidation does. If you read your material, you're going to see this. Two carbons. So it's a cycle acting on two carbons in a cyclic rotation, keeps going until the whole fatty acid is exhausted. And you have from 16 carbon chain fatty acid to 8 molecules of acetyl-CoA. The third oxidation is catalyzed by beta hydroxyacyl-CoA dehydrogenase, which gives you this. And then the last reaction, the thiolysis, which adds CoA SH. S-sulfur, SH, reduced sulfuric group. Basically is what causes the name here, thiolysis, if you still remember your chemistry. And this reaction is catalyzed by acyl-CoA acetyl transferase. And that's where you end up with your acetyl-CoA and another molecule. This now will have 14 carbon chains because 2 carbon chains have been removed as acetyl-CoA from the original 16. And now this is beta oxidation. And this acetyl-CoA will now go into your, what do you call it? Your TCA, citric acid cycle. So basically what I was saying here has been elaborated here, right? 16, beta oxidation will remove two carbon molecules identified as acetyl-CoA. From 16, you have 14. From 14, you're going to have 12 because of beta oxidation. Another cycle of beta oxidation will give you 10. Another cycle of beta oxidation, 8, 6, 4, and then the last two. That's it. All right. Basically, this is beta oxidation. And then you need to calculate the ATP yield you're going to get from the cycles of beta oxidation. And when these go into the electron transport chain, how many molecules of ATP are you going to get? This is quite self-explanatory. The acetyl-CoA, the hydrogenase will give you, because from 16, you're going to have 7 cycles. You're going to get 7 FADH2, 7 NADH, and then if you go into the citric acid cycle, you're going to get 8 NADH. Alpha ketoglutarate will also give you 8 NADH. Succinate dehydrogenase, 8 NADH. Malate dehydrogenase, also 8. So if you sum everything up together, I think you should get this total. Okay. So this is for module 2, session 1, which I think is a continuation of your fatty acid oxidation. I think more or less we've done justice to this topic. It's up to you now to read, and you drop questions in the forum if you have any questions, or you're having difficulties grasping a certain concept. Please feel free to contact me via the forum, of course. Okay, so I think this uh, should be the end of module 1. That's session 4 of module 1, basically. All right.